This presentation follows a study that found that white adipocytes and subcutaneous fat depots require KLF15 for maintenance in preclinical models. To introduce the topic, an easy question to ask is, does someone in your life struggle with obesity? This study shows promising solutions for the obesity crisis using cells our body already produces. According to the CDC, 2 in 5 adults are obese in the US. Adipose tissue, which makes up body fat, is composed of cells called adipocytes. The two types of adipocytes, brown and white, have different roles in maintaining energy in the human body. Brown adipocytes are present at birth and are used to maintain body heat in babies by burning energy. Brown adipose tissue, or BAT, could be helpful in addressing obesity as it is used to burn energy, but the issue remains that humans are born with more white adipocytes than brown. However, recent studies have shown that some adipocytes can have characteristics of both white and brown adipocytes, making them more beneficial for burning energy. This study investigates what factors need to be present in order for these white adipocytes with brown characteristics to stay in the body. Grupo-like factors, which are zinc proteins used to regulate metabolism, are hypothesized to be a key factor needed in utilizing white adipocyte tissue characteristics.
All right, y'all. So we've got this experiment here where we are trying to better understand the chain reactions within a cell, within a fat cell, that tells it, hey, it's cold. We need to increase the heat. So we're trying to understand that mechanism. And there are some practical methods that we used in this experiment to try to figure that out. So we're going to go over those steps now. But they did it in 4 main ways. And I really like how they did it because we zoomed out and then we zoomed really close in. And so now I'm going to break down what those experiments looked like. Mice analysis. We've got 2 groups of 3 mice, male and female. We have normal mice, but then we have our genetically altered mice, where we took the mechanism in their genetics that says we need to make our bodies warmer when it's cold. We removed that from their genetics. And then, okay, this is where if you're with PETA, you want to close your ears, but we leave them at this 39-degree temperature for several hours. And then we start measuring the body temperature of these 2 groups. Now, keep in mind, one of these groups of mice has had the genetic mechanism within them that tells their fat cells to produce heat. That has been removed. All right, y'all. So now we're looking at cell analysis. So we've gone from rat all the way inside to the cell. We have these cells that are completely removed of their ability to produce heat, and then we give them drugs that tell them to produce heat. So if those cells produce heat after we have removed everything that allows them to do so, then there's something we don't understand about how cells have a chain reaction that tells them to produce heat. So those were the mouse cells. Then we put our human cells. And, human cells are a little harder to work with than mouse cells. So we actually presented a virus. A virus is just a bunch of genetic material inside of a protein coat that we made, and then we injected into them. And that sounds so scary, but it's kind of like an update on your computer. We put this information into the cell, and we tell the cell, hey, if you ever feel like warming up, just go ahead and cut that out, is what that virus does. So we did the same thing. We give that human cell medicine to increase metabolism, and then we just measure the temperature. And then we are going even deeper, as deep as we really can go with genetic material analysis. So we've mashed up the fat cells of the normal mice, the gene-edited mice, and the viral cells, and we've collected pure samples of RNA, which is a really basic form of genetic material, from each of these guys. And then we're using this piece of technology. It's called a plasmid, and it really allows us to take RNA and mutate it to make DNA do what we want it to do. And so we make DNA where the heat-making gene is turned off. However, we attach a glowing green device to the gene that makes heat. So it's still within the cells. We didn't rip it away. We just turned it off. And so if this DNA that we've created, if it still produces heat after all these measures we've taken to prevent it from doing so, then there's something we don't understand about the mechanism.
So as for our results, first we established that KLF15 is basically the off switch for fat burning. We found that natural brown fat has very low levels of KLF15. When we genetically deleted KLF15 from white fat cells, they underwent a transformation called Beijing, as you can see on this chart. The genes responsible for burning fat, especially UCP1, they skyrocketed. And this happens because without KLF15, the cells build bigger ADRB1 receptors, and these are receptors that are hypersensitive to signals telling it to burn fat. We then looked at what happened in the live mice, and the results were visible. If you look at the top photo on the right, it shows normal white fat, which is more pale, while the bottom photo shows the fat from our KLF15 deleted mice, and it's physically darker and more redder, and that's because it's becoming beige fat. Functionally, these mice become super burners, and the graph shows their energy expenditure. The mice lacking KLF15 burn significantly more calories per gram of body weight, and they were also better at maintaining their body temperature in a colder environment. Importantly, they weren't moving around more, their fat was just working harder. Finally, we had to answer the most important question. Does this work on us? Does this actually work on humans? We confirmed the exact mechanism. KLF15 binds directly to the DNA to block the production of the burn receptors. When we tested this in human fat cells, we saw the same exact result. Human cells with lower KLF15 levels consumed more oxygen and became much more sensitive to weight loss drugs. This proves that targeting KLF15 isn't just a mouse experiment, it's actually a real potential pathway for treating human obesity as well.
The major takeaway the researchers were able to identify two genes in fat cells that control their characteristics, KLF15 and ADRB1. KLF15 is a gene, and all genes code for proteins. KLF15's protein is a transcription factor. Transcription factors are proteins that are able to bind directly to DNA, controlling the expression of certain genes. In the case of KL15, it binds to the area of the DNA that controls the activity of ADRB1. KLF15 is a repressor transcription factor, so when it binds upstream of the ADRB1 gene, ADRB1 expression is low, so very little ADRB1 protein is produced within the cell. The ADRB1 gene is a really important gene in brown adipocytes, the fat cells that help us break down lipids in the body. The ARDB1 gene encodes a protein that is essential for performing lipid or fat metabolism. In white fat cells, which are the fat cells that store lipids and overall are bad for human health, KLF15 levels are high, which means that it is bound to the DNA and ADRB1 levels are low. This means that white adipocytes or fat cells are unable to perform lipid metabolism, meaning they store lipids in our bodies, which can have negative health impacts. These findings are extremely important for understanding fat cells and how they maintain their characteristics because they show that the characteristics of fat cells and their maintenance is dependent on gene expression. And this is important because gene expression can be manipulated. The researchers were able to manipulate fat cell characteristics by completely knocking out the KL15 gene in white fat cells in mice. By knocking out the KL15 gene and effectively getting rid of the KL15 transcription factor, nothing was silencing the ADRB1 pathway, and so that protein, which is essential for lipid breakdown and metabolism, was being produced, so these white fat cells were able to perform the function of brown fat cells. Effectively, the researchers transformed the dangerous white fat cells that store lipids into brown fat cells, the healthy fat cells that are essential for breaking down fat in the body. While similar pathways for fat metabolism and fat cell maintenance have been found, there hasn't been a pathway found that is conserved or the same in humans. The researchers of this study found that this pathway, the KLF15 ADRB1 pathway, is conserved in human fat cells, meaning it is a prime target for human obesity intervention since gene expression can be manipulated. Moving forward, this research lays the foundation for translational research, which is when scientists take understanding of the way our genes and our cells work and transform that into intervention and treatments that can help human people and human patients with the diseases they struggle with. Identifying this pathway, KLF15 and ADRB1, allows for researchers to begin to think about and develop techniques and interventions that may regulate or modify the expression of these pathways. This presents an opportunity to target diseases like obesity, like insulin resistance, things that stem from having high levels of unhealthy fat cells like white fat cells in our bodies. The implications this research has for obesity and metabolic disorder treatment is great, as it presents the opportunity to transform dangerous white adipose tissue into less harmful beige fat cells, which could minimize the adverse health effects of excess adipose tissue.